Okay, good morning, good afternoon, good evening. Welcome to the MechE Alliance seminar series. Again, my name is Brian Anthony. I'll be your host for today, really to introduce our speaker, and then at the end, my job will be to marshal the questions that you'll ask to Professor Traverso. So this is being recorded. Please, at the end, raise your hand or use the Q&A function to send your questions. So it's my pleasure to introduce Professor Giovanni Traverso, who's going to share with us today his very interesting work in gastrointestinal device development. He has actually a rich background in engineering and in medicine, and brings to bear both on what he'll be sharing with us today and in other device work and other ways to address COVID and other very exciting areas. Gio, I thank you for your time and look forward to hearing about your exciting work, and take it away.
Thank you so much, Brian and Teresa, for inviting me today. And let me see if we can move forward. Teresa, can we? I'm not sure if we can go to my slide.
You should just go ahead and share. Yeah.
Share. Okay. Great. Okay. Well, thank you so much. So, the focus of our group really, I would say, is largely around the development of systems that reside in the GI tract. And so, as Brian alluded to, I'm, in addition to sort of an engineer's hat, I also wear a clinical hat, and I'm a practicing gastroenterologist at Brigham Women's Hospital. And so, certainly, I think, and I also spend time within the GI tract, helping people in different ways. But certainly, from an engineering perspective, really thinking about how we can develop systems to make it easier for patients to really adhere and comply essentially with our therapy to really maximize their opportunity for health. And so with that in mind, a lot of what we do centers around developing systems that make it easier to take medication or sense different conditions, and really try to bring those together to bear on the health of the individual. One of the core aspects that we try, or challenges that we try and address, is really based on a very simple observation, and I think we all recognize this, and that is that drugs simply don't work if you don't take them. But in spite of this sort of obvious statement that was really, I would say, popularized by a former Surgeon General from the 80s, and this is C. Everett Koop right here, what we recognize is that as a whole, as a population for chronic therapies, folks in general only take medication as prescribed 50% of the time, which is just, astounding. But that lack of adherence or imperfect adherence really manifests in tremendous effects on societies, mainly morbidity and mortality, and therefore significant costs to our medical systems. Just to give you a sense on this problem, this is an editorial from the New England Journal a few years ago talking about this problem. And what we recognize is, that this is an enormous problem in the US, where there are expenditures in excess of $100 billion annually attributed to non-adherence. And what we appreciate is that if we were to address adherence, for example, for high blood pressure, for hypertension, we stand to mitigate or prevent the premature deaths of nearly 100,000 individuals alone per year. And so, what we recognize is that non-adherence or non-compliance is really a challenge of epic proportions. And so anything that we're able to do here can really have a significant impact. And there are several wonderful pieces that have been written about adherence over the years. There's additionally a very nice report from the early 2000s for the WHO. One of the things that report talks about is the impact that addressing the non-adherence problem could have on society. And what's been recognized by the field is that improvements in adherence actually stand to enhance our capacity for health even greater than the introduction of new medications in many different diseases. And so as engineers, what we started to think about was, well, how can we help this problem? Is there an observation? Is there something that we might be able to wrap our heads around and try and help people? And there's a very simple observation, which is if drugs are dosed more infrequently, one is more likely to take them. So this is data taken from a review in the New England Journal on non-adherence from several years ago that looked at medications that are dosed at different intervals, whether it be once a day or 4 times a day. And perhaps not surprisingly, when drugs are prescribed once a day, an individual is more likely to continue with that therapy. So that, I think from personal experience, at least that's been my experience. The times I've had to take something every 6 hours, like certain antibiotics, it's a pain. And I typically don't take them at the right time, or I miss a dose. And this certainly is borne out in clinical trials. But what is interesting is actually you can continue to observe this trend. And so if you look at longer term data, and this is for a class of drugs in the area of osteoporosis or treatment of osteoporosis. There's a class of drugs, they're called bisphosphonates, that are used for the treatment of osteoporosis. And what we see is that if those drugs are prescribed daily, weekly, and monthly, and I should add that this is one of the few drugs that today exist in those formats, what we see is that in general, those drugs that are prescribed more infrequently are ones where we observe persistence or adherence rates for longer periods. And so with that in mind, what we started to really think about was, well, could we develop a system that enabled oral delivery of a system that provides continuous or controlled release over a long period? And so here I'm showing you a cartoon of this basic concept. This is a cartoon in The Economist about some of our early work in this area, but I feel really captures what we're trying to do, right? Which is have something that is dosed orally and that provides metered dosing over a long period. Obviously, hopefully in a smaller form factor than what you're seeing here in the cartoon. Now, the challenge with this is that the GI tract has this incredible capacity to transit material. And so if, as we all experience in general, when you ingest something, it typically transits out of your body within a day or so. And so the question is, well, how do we overcome that challenge? And certainly this is something that the entire field has been considering now for many decades. So when we think about a standard capsule system, that typically goes down the esophagus, through the stomach, and then the small intestine. Typically, most drugs are absorbed in the small intestine, and those tablets or capsules and the materials will get to the small intestine within a couple of hours, and the drugs would be absorbed then. And some of the ways in which people have tried to approach this is by adding elements that, for example, increase the residence by mucoadhesion. So having systems stick to the GI tract. And certainly that can increase the residence and how long things take to actually transit. And that can give you an extra couple of hours. But really what we want to do is how do we achieve ultra long release? And so our approach has been one where we have systems stay in part of the GI tract. And the area that overall we've focused on has been the stomach. And what you see there in the cartoons at the bottom are essentially the drug release kinetics that we're hoping to achieve, where we have the sustained stable level over a long period. Now, whenever we think about these systems, I think it's important to think about sort of the basic steps and how we think about the challenges for each step. So if we're proposing for someone to take something orally, the first thing we need to consider is the capsule, what is being ingested. And you see a standard capsule here. This is a gelatin capsule. And so we have a folding or a packing challenge or problem in front of us. And the reason that we need a capsule is that we need to get through the esophagus. So this is really a ship in a bottle kind of problem, right? Which is how do we get a ship through the neck of the bottle and then expand or unravel and change in the stomach? And so, that was really, the first challenge that we started to consider. And we looked at many different shapes and approaches to do this. And the next challenge is, once you're encapsulated and you get to the stomach, you need to deploy, you need to unravel. And so here you see a video of a stomach of a pig showing one of those very early prototypes, unraveling. Now, once it's deployed, we need that system to stay there. But not only do we need it to stay there, we need it to provide a continued and controlled drug release. We need it to be able to maintain the drug stable in a relatively harsh environment. And then eventually, whatever is being retained needs to eventually pass somehow. It needs to break up in a controlled way. And so whenever we're developing new systems, safety really is paramount, particularly early on. And so one of the things that we started to consider and really brainstorm around were, all the different ways in which these systems could cause problems. And one of the primary ways that we recognize is that if one of these systems passed into the small intestine, which is a lot smaller and narrower than the stomach, it could cause a blockage. And so we needed a way of having these systems dissolve preferentially in the small intestine. So one of the very early things we did was actually develop a material that we refer to as an enteric elastomer. And let me explain what that is. So we wanted a material that would be stable in the more acidic environment of the stomach, but would degrade in the near neutral environment of the small intestine. And we wanted a material that could bend, that would have some flexibility. And so hence the term enteric elastomer. So the term enteric refers to that ability to be stable in an acidic environment, but unstable in a near neutral environment. And then the elastomeric component, as you can see on the right of the slide, provides the flexibility. And I'll show how that plays into the development of the dosage form. What you see on the left are dissolution kinetics of the material, either in an acidic environment like simulated gastric fluid, which is SGF, or in a near neutral environment, which is SIF, or simulated intestinal fluid. And so you see that in that environment, the material starts to break down. Now, using this material, we were able to generate some of the earliest prototypes to start iterating in a large animal model on a whole range of parameters. But let me walk you through some of the early things that we did. We created molds out of silicone, and what you see up here in the top panel or row are intervening elements of polycaprolactone, which is a commonly used polymer. It's used in cups and used in a whole range of different applications, both in the biomedical space as well as consumer space. But additionally, we added the enteric elastomer. And this allowed us to take this rigid polymer with intervening flexible segments, fold it up, and put it inside of a capsule. Then we place those circular prototypes either in simulated gastric fluid or in simulated intestinal fluid. And what you see is that the shape that was designed, or I should say imparted into the structure, could be assumed again upon coming out of the gelatin capsule. But if placed in simulated intestinal fluid, the circle would break up over the course of hours. And so with this and with these tools, we were able to start iterating now in different designs. And so we started looking at Starlike systems. And what you're seeing here are serial x-rays of a live pig. So the questions we were started to ask were, well, how long does it take for these systems to unravel? And how do we optimize these systems to unravel and open up fast enough so they will stay in the stomach? And so we developed Starlike systems and we developed hexagonal-like systems. Now, this was reassuring, but we wanted to leave these systems for longer periods. And what you see here is one of those early prototypes that's been now in the stomach of a pig for about a month. And the couple of things I want to highlight here are that the stomach is empty. These systems are not causing any kind of blockage. And two, the mucosa, the tissue, is pink, it's healthy. There are no abrasions, there's no blood. And so those were really reassuring. And we did many experiments in pigs at the time. And pigs are an interesting model because their GI tract, it has similar dimensions to that of humans. And so that was really helpful to allow us to really help convince us that we were on the right track as far as developing a system that could reside safely without really affecting the transit of food. And I should add that these animals, these pigs, were on a very liberal diet that included a lot of indigestible materials as well. And so some of the questions that I sometimes get are, do things get hung up at the level of these systems? And, in these animals, I mean, they eat everything from whole bananas, including the banana peel, to raw sweet potatoes, to, the feed. And so it was very reassuring for a fairly, diverse diet, at least, that we weren't seeing any collections forming or any blockages. Now, this was all well and good, but really what we're trying to do is provide extended drug release. And at the time, we were working with the Gates Foundation, and they were interested in a drug called Ivermectin. And so Ivermectin is a drug that treats parasites. The work behind Ivermectin was recognized with a Nobel Prize a few years ago for its application in the treatment or eradication of river blindness in sub-Saharan Africa. What is interesting about Ivermectin is that in addition to being able to help eradicate river blindness, it has an effect on the mosquitoes that carry malaria. And so during the river blindness studies, there was an interesting observation that was made that as those campaigns of treatment would go through a village, what was also observed was that the mosquito burden happened to also decrease. So interestingly, ivermectin has multiple effects. It can help treat a whole range of parasites, but also is toxic to the mosquitoes that carry malaria. And so the Gates Foundation came to us and said, could you help us develop a system that could provide more sustained levels of ivermectin? And so this is actually the very first set of experiments that we did with ivermectin. And what you see on the bottom panel are basically the drug release kinetics of a standard capsule of ivermectin. Typically, you want to be above the level that's noted there in the red dotted line. And you have that level for about 2 days or so in the standard capsule. Initially, we got out to 10 days with a STAR system. And actually, by the time we got to the publication, we were out to 2 weeks, which was very reassuring. So this is really the first proof of concept that we can have a capsule that stays in the stomach for a prolonged period and can provide sustained drug release in a large mammal. Now, we had settled on the STAR system, and there are a couple of things that we recognized very early, and those included the fact that each arm is an individual component, and so that can be considered either for an individual drug or can be even subdivided for different drugs or different formulations of the same drug. And formulations refers to drugs where we add different additives that can help tune how they're released. And so one of the next challenges that we tackled involved HIV. And so we recognized that if we were to develop a system that could provide drug, let's say for a week, we could help make it easier for patients to receive their HIV medication. There are other ways in which these systems can be used, which is also to protect people against HIV. So people who, for example, are or work in high-risk situations where they may contract HIV. And so we aim to develop the system specifically for antiretroviral drugs. So we looked at all the drugs that were available at the time, and we identified these 3 drugs as ones that were potent enough to fit in a single STAR system, so in a single capsule, to provide treatment for a whole week. And then we were able to develop, I'm just going to show you one example here, a system that provided this, again, sustained release from a single capsule. And this is data, again, in pigs. And we have sort of the immediate release control, which is in red, but then the sustained release, which is the STAR system. So again, this was very reassuring, again, validating the capacity of the system to do this. Now, more recently, another big area that the Gates Foundation, this is work we've been doing with the Gates Foundation now for about 7 years, they recognized, an opportunity for potentially helping treat, or not treat, but rather help women with control of fertility. And so in this case, their interest was, well, could we do it for, essentially develop a once-a-month oral contraceptive system? And so that's actually what we did. And we published this last year. And what we showed was that using a single STAR system, we could achieve the levels required that would cover, somewhere between 3 to 4 weeks of dosing of a contraceptive drug. So that was, I think one of the key areas. We continue to work on systems that make it easier. There's a company that we started several years ago that is translating and is now evaluating several of these systems in humans, and there are multiple clinical trials that are ongoing. Now, let me transition to another area, which really, is another big area that our group really continues to work on and really tackles from multiple different angles. And let me start with 2 clinical cases. I'm going to switch a little bit to more clinical realm. And these are 2 clinical cases from, actually, these both are patients of mine from a long time ago. They're fully de-identified, but nevertheless, they, I think, provide a really helpful teaching point. So this was a really memorable case for me. I was a young fellow, meaning a clinical fellow who was learning about gastroenterology at the time at the Mass General. And I was called to see someone who was a prisoner at a local jail. And I was called by the emergency department physician who told me that this person had ingested shards of glass. And so I went down to see this patient, and they were sitting, and I'll never forget this, they were sitting in the emergency department. They had a handcuff on one hand to the chair that they were in, but with the other hand, they were holding a sandwich and they were eating a sandwich. And this was really interesting, and it really, I would say very formative part of my gastroenterological sort of training experience. And one of the things that we recognize as gastroenterologists is that if someone has eaten recently, we are unable to conduct or perform a procedure safely because there is a risk that the food can come up and go down the wrong way. And so one of the things in evaluating the patient that we could appreciate was that the shards of glass were about just under 2 centimeters in size. And what we know is that sharp objects that are under 2 centimeters generally pass through the GI tract without causing problems in the majority of cases. And so, one of the things I say, and this is really based on the literature and some of the references there at the bottom of the slide, is if everybody on this call, and I'm not suggesting anybody do this, but if everybody on this webinar were to swallow a razor blade, 80 to 90% of you would be okay. Again, I'm not suggesting you do that, but that gives you a little bit of a sense of our GI tract capacity to tolerate the sharp objects. And so what was really interesting to me here was really observe this patient over the course of a few days. And, those shards of glass passed, and the patient, really was none the wiser as far as when they passed, but really taught me about the capacity of the GI tract to tolerate sharp objects. Now, the second case is one that is perhaps a little bit more common in gastroenterology. And this was a lady in her 60s with a history of osteoarthritis who had been taking a lot of ibuprofen, Motrin, which many of us take. It's a wonderful drug, but it does have some side effects, and it can lead to ulceration in the GI tract. And it can be so severe that it can lead to bleeding. And in this case, this patient presented with epigastric pain. Epigastric pain means pain here at the top of your abdomen, which is where your stomach is, and black tarry stools and lightheadedness. So black tarry stools is a sign of bleeding in the GI tract because blood, when it gets digested, turns black. And so that is a very classic sign of bleeding from the GI tract. And lightheadedness in the context of black tarry stools and pain in the abdomen really signals blood loss and, makes us think typically of, a bleeding ulcer. And so in this case, given this history of significant ibuprofen intake, coupled with a physical exam, which had epigastric pain, and a history that provided black tarry stools and lightheadedness, our index of suspicion for an ulcer is very high. And so an endoscopic procedure was performed, and what we saw was an ulcer in the stomach that was bleeding. One of the things that we do to help get control of the situation when we're doing this is that we inject the ulcer, and we inject it with epinephrine or adrenaline. The reason that is because epinephrine or adrenaline, what it does is that it induces a local response. And it induces specifically what's called a local vasoconstrictive response, meaning the blood vessels, the arteries, will tighten, and therefore the bleeding will stop. And what that allows us to do is really visualize that ulcer bed to see and then treat it. And we treat it with little clips, or we can cauterize. And so, but as soon as we inject that adrenaline, what we notice is that the heart rate goes up. And that is because of a systemic response, not a local response. So that injection in the wall of the stomach is going into the circulation and then up to the heart and stimulating the heart to beat faster. So that observation really tells us that if we use an injection, we have the potential to reach the bloodstream very rapidly. And this is something that all gastroenterologists recognize, is that as soon as we give these injections, usually there's a heart rate monitor that is bing-bonging in the room, is going crazy. And so putting these pieces together, what we wondered was, well, could we develop ingestible systems that gave injections from the GI tract? And this was about 8 years ago now, 9 years ago, when we started. And so, we put some basic, concepts together, and we made some prototypes. And here I'm just showing you some of that early work where we made, some basic CAD drawings and made some prototypes, fed them to pigs. We then injected insulin in different parts of the GI tract. And what we saw when we injected insulin in the stomach, in the small intestine or the colon, was that actually we could lower the glucose. And we could achieve comparable levels of lowering to that is achieved through injection across the skin. So this really was motivating insofar as trying to do this, but it left many questions open. And specifically, there were three things that were open. One was, how do we get these systems to interact with the tissue, with the mucosa, in a reliable way? How do we trigger the injection or actuate that event and have essentially that injection occur? And then how do we inject a sufficient amount of drug to actually have an effect, given that we want systems to be small so they can go through the GI tract safely? And so let me walk you through on how this was approached. So as far as engagement with the tissue, we took a page from toys like Weeble Wobbles, but also self-riding tortoises, where by having a density difference and a specific shape, we could ensure that using gravity, we could ensure we knew that the bottom end was in contact with the tissue. And this is a concept that we tested through high-speed photography. We compared it to other shapes, whether it be ellipsoids, spheres, and we looked at it we evaluated the performance both in vitro in different fluids, but also inside of pigs, and simulated their walking or movement to really observe, this self-orientation occurring inside of a gastric cavity, inside of a stomach. So that was, that was sort of the first piece, as far as trying to ensure that we had that juxtaposition of the business end with the tissue. The next thing was, well, how do we get these systems to trigger? And there, we really wanted to think about cues that are universal to all of us. And so the cue that we identified that really is universal to anyone on the planet that is alive is humidity, in that the gastric cavity is a humid environment. And so using that, we wanted to identify a material that would break up that displays brittle fracture mechanics. And so here, the way I typically describe it is, think candy cane, not taffy, right? So you want something that gives way very suddenly, as opposed to slowly stretching, right? And so this is where using sugar glass, we're able to do that. And we showed both through modeling, but also experimentally, that using sugar glass, we were able to have very tight control on the release. And just to give you a sense, here we have one of those early prototypes of a spring encased in the sugar glass and in a humid environment. And you can see that sudden release of that stored energy that occurs all of a sudden to really support the injection. Now, the next challenge was, how do we get enough drug inside of the body? How do we inject enough, given that what we want to deliver is a sizable blueberry? And typically when we think of insulin, we think of a solution. And the issue with solutions is that they are limited generally by the solubility limit. And so typically when you have a solution of a drug, you don't want to see sediment at the bottom, you want a clear solution, ideally, or a homogeneous solution. And so what we ended up really focusing on was solid formulations. And so what you see here in this slide at the bottom right is that prototype of a solid formulation where we made up to 100% insulin needles. So we're actually pushing into the tissue these tiny solid posts of the drug, and then inside of the tissue, they dissolve and go into circulation. Now, we did all this, and then we showed that they worked in a pig. And what we showed was that the levels of drug that we could deliver were comparable to the levels of drug achieved when administered through the skin. We also did a lot of safety experiments ensuring that we weren't going through the tissue, both in the stomach of pigs, but also cadaveric human stomach. So where we are now is we have a system that can self-orient, senses humidity, uses that cue, and then triggers an injection event. And we have the capacity to deliver a range of drugs. Now, as we sort of think about, delivery science, one thing that I think is really important is, how can we do better? And so when I think towards closing the loop, one of the things that we recognized early on with our systems was that, they could reside in the body for a long time. What if we added sensing capabilities to these to really amplify their capacity to treat a patient? And so, but as soon as we start to think about that, the challenge that we start hitting up against is, well, how do we power these systems for prolonged periods? Like if we're trying to keep these for a month, let's say, how do we get enough energy? And so here, we took a page from our high school textbook, and what we created here is our galvanic cell system. So here we have a standard lemon battery, and this is something that I've done with my kids recently during the shutdown. It's a fun experiment. But we did something similar in the GI tract. In this case, we looked at different types of electrodes and ones that were stable in the environments that we're interested in tackling. And the ones, the pair that we identified were zinc copper electrode pair to the demonstrate stability. And in general, I think what we recognize is magnesium is certainly good, but it'll dissolve very quickly in these environments. And so having identified the electrodes, we created characterization systems. So we created a capsule that could evaluate essentially the peak power that could be harvested from the stomach and the intestines. Interestingly, we observed that certainly we could identify that in the stomach, and surprisingly, we also observed that in the intestine, even though it's not an acidic environment as the stomach is. Now, with this information, we then created a prototype and that we gave to pigs. And this is a prototype that is fully powered by the harvested energy, and basically where we were sensing temperature and we were communicating that to a base station. And we showed that we could keep this system viable and functional for about 7 days. And so, at least now where we are today is that we have the capacity to power through harvesting, but also using some sophisticated batteries. And so now a lot of the work that we're doing really is around, well, how do we enhance ingestible systems to not only deliver, but sense and potentially couple the sensing to the delivery and communicate different phases of health and disease. Now, with that, it really takes many, many people to do all of this. And I want to acknowledge the efforts of many of the folks that have been instrumental in these efforts. And so for the STAR system, I want to acknowledge Ameya Courtenay, Andrew Bellinger, Chi Zhang, Bob Langer, Philip Eckhoff, Anna Slater, Allison Hill, Johanna Daly, who really represent collaborators and the leading scientists, some of whom were at MIT, some still at MIT, some at the Gates Foundation, some were at Imperial, but really all over the world, really helping realize the work behind the STAR. On the ingestible microneedle system, this has been a long, relatively long, I would say, effort that's been now going for about 9 years or so. And this is work initially that I did in collaboration with Carl Scholhammer, and then subsequently Alex Abramson, Essa Cafferel made significant contributions, as well as Nicholas Rockstead and our collaborator in industry, Ulrich Rabeck. On the harvesting systems, we continue to work on ingestible electronics, and this is work we did in collaboration with our current dean, but also it was led by Philip Nadeau, who is a former graduate student in EECS. But really, again, I think it's important to recognize I'm the one here presenting. It's really a village. And these are some of our folks before the shutdown a couple of years ago over in the Cape. And with that, I want to thank you for your time, attention, and I'm happy to take any questions now, but also leave my email and feel free to email me with anything. And of course, I want to acknowledge all the wonderful sponsors and continued support.
Yeah, thank you very much. I send you the physical and the virtual clapping. And that was phenomenal. So participants, please use the Q&A function to ask questions. But let me just ask a question to seed the conversation here for a few moments. As you look at these various technologies, Gio, I was wondering if you could share sort of the path that's ahead of you to sort of look at how do you translate these things to a device. What does that journey look like from where you are now?
Absolutely. And I think, we certainly think about very much around the full arc from starting in the lab all the way to the first in human and beyond. And what we recognize is that we need to work with partners. Just as we have collaborators in the lab, we have collaborators on the biotech or industry side. And so, for example, the STAR system work, that's work. We started a company 5 years ago now called Lindra, and they have an incredible team that are really doing transformational work. I mean, what we do in the lab is, very early, and the challenges in manufacturing, scale up, regulatory aspects, execution, I mean, they're just enormous. And so it really takes extremely talented people who are 100% committed to getting these systems through. So that is a key element. I mean, on the ingestible system for drug delivery of insulin, for example, that's work that's been in partnership with Novo Nordisk. And Novo Nordisk is a fairly big pharmaceutical company and the #1 producer of insulin in the world. And again, I mean, it's been an incredible collaboration and continues to be to try and get these systems into humans. On the electronics front, it's an earlier set of programs, but we certainly continue to work with a range of collaborators to get these systems forward. So I think, if anything, I think collaboration with biotech and industry are really essential, at least in my experience, to help accelerate and introduce the systems into the market. And I think, as an investigator and as an academic researcher, I'm very passionate about moving things out of the lab. And I think, there's nothing like the investigators and the researchers being involved in that process. And I think it's critical, if we want to see our systems have an impact, to really continue to participate beyond just publishing a paper or submitting a patent. And sometimes, it's work that perhaps isn't recognized, but I feel that the impact is what's recognized. And I think that if it's not even the recognition, but rather is extremely, I think, satisfying to see systems, move forward into helping people. And so, again, spending the time on that aspect, engaging with the licensing office, with potential partners to license the technology, and then fundraising to continue the work. I think being an active participant in the whole process, I think, is critical.
Great, thank you for that. Participants, please use the Q&A if you're comfortable. And I also received a direct chat here. The question was, how does this help us in COVID, in addressing COVID in some way? And so I'll let you answer that, Gio, but sort of you may want to bring in some of the other work, you mentioned the other work that you're doing that is in that space.
Yeah, no, absolutely. So, certainly I think for the different drug delivery systems, they might be coupled to drugs, whether it be, some of the ones that are being investigated. I mean, during the shutdown, our group actually has been very active across several programs to tackle COVID. And so we've had efforts around ventilator splitting. We've had a very active effort on injectable masks. So these are injection molded masks that are reusable and sterilizable, where we've done our own trials, and and really pushing forward to help provide a solution that is scalable. We've also collaborated and have an active collaboration with Boston Dynamics to really amplify robotic enabled telehealth in emergent settings to facilitate and mitigate the risk of exposure to healthcare professionals, as well as potential savings on the protective equipment side of things. And so, what I at least recognize is that I think as engineers, we have the capacity to really help across many different, I would say, avenues to help people. And I think it's just a willingness to engage in that and really collaborate. And I think all of the COVID efforts that we have are really through extensive collaboration. And I think we're extremely fortunate to be in the city where we have, some of the best hospitals in the world, some of the best universities in the world. And I think from a biotech and pharma perspective, again, several of the leaders in the world. And so I think it's really reaching out, not being shy, and I think that's at least how we've approached the COVID situation, and really trying to help as much as we can, and being willing to engage in that dialogue with potential partners and collaborators.
Thank you for that, Gio. We have a question here. We have a little bit more time. I'll just ask it here. Does the consumption of more basic foods on the pH scale meaningfully affect the dose rate of the delayed delivery systems?
So, I'm not aware of data. I mean, our bodies are extremely good at controlling pH. So I'm not aware of significant variations that have on the luminal pH as a function of diet. But I'd be, if the person has some examples, that please feel free to email me. But but again, I mean, our bodies have such an incredible capacity for buffering, and one of the things that we see clinically are very small changes in pH mean that you're very close to death. I mean, it's what we see clinically. And so that speaks to the incredible capacity of the body to really buffer the different environments. Now, I think one thing to add on though to that, to that question, and I think it's an important regulatory question, which is how do different ingestions, and very specifically fats and alcohols, are typically the areas that are really fall under the regulatory aspect. So typically there are certain conditions that have to be evaluated for different dosage forms. And those include diets that have a high fat content, because certainly certain drugs can partition into fats, for example, and fats themselves can help release or accelerate the release or delay the release depending on the system. And alcohols too. Alcohol, as a solvent, can also help facilitate that release. But on the pH side, I just haven't seen as much, and I'd be curious to know what But so feel free to email me, and happy to discuss that further.
I want to thank you again, Gio, for your time. Send the physical and virtual clap here, and commend you for all your work, both in the gastrointestinal tract and helping out with COVID. So again, thank you. And participants, thank you for your time. Teresa, if you can bring up the exit slide. Normally our schedule is once a week on Wednesday, but as I think many of you know, we had to postpone our talk from last week due to the Shutdown STEM initiative. So we will have an alum from the department, Ed Bernardin, who will be talking about the World Rally Cup and autonomous vehicle technology tomorrow. So same channel, but tomorrow, Thursday, at 12 noon. So I hope everybody here can join us again tomorrow for the next MechE Alliance Seminar Series. And thank you again, Gio, for your time, and everybody for sticking around. Have a good day.